Our speaker today is Thomas Zinnen. Professor Zinnen has been a biotechnology extension specialist since 1991. He also served as a Congressional Science Fellow from 2000 to 2001. He was on loan to the National Science Foundation from 2008 to 2010 as a speechwriter for the NSF Director. He leads BioTrek, the public outreach program of the Biotech Center and UW-Extension. His key role is in welcoming Wisconsinites to come and experience science at their public land-grant research university in Madison. Please welcome him. He's going to be speaking today about the changing colors in the spectrums of biotechnology and food. Welcome, Tom.
Thank you very much, Julie. Thank you for the invitation. It's great to be able to come to Wausau. I've spent many hours in the room next door with 4-H STEM meetings, science, technology, engineering, and math, but I've never been in this auditorium. It's going to be a good time. I know Julie asked you to silence your phones. I've turned mine on. Holy cow, it's 8:54? You blew through that. That's great. Well, you get to have the 2.5-hour version of my talk. I understand I'm supposed to be done around 9:45, 9:50, but since I get to start early, I think it'll be all right. I'll just try to keep track of how much time I'm using. Whoa, that's me. I'm from Dixon, Illinois. This woman right here had a roommate from Dixon, Illinois when she was in college at Northwestern, and they didn't get along. I almost laughed when Julie said, the whole state of Illinois shuts down from Christmas to New Year's. It's like the whole state of Illinois shut down, period. So I work for UW-Madison. I also work for UW-Extension. I went to undergraduate school at UW-Platteville. I am from Illinois. I understand what it's like to have a competent state university system, and not because I grew up in Illinois. It's good to be here at UW-Marathon County. Today is the 10th of January. Did I get the date right? Whew. All right, let's see how this works. Boy, is every slide going? Oh, okay. This is why I like my job. I have a job because of bovine growth hormone. And some of you remember bovine growth hormone. It's my favorite little thing. Remember 1989 and who was the governor then? Tommy Thompson. You just saw his picture up there because he's going to be speaking. And who was the state senator then, about 30 years old? Feingold. And what did they do in 1989 and 1990? They had a little thing about bovine growth hormone. The state legislature passed 2 bills affecting bovine growth hormone. The governor signed one, and the one that the governor signed included language to set up the program that I have and that I started as an employee in on June 1st of 1991. Biotech and food is profound because it affects so many basic parts of our lives. So it's not just a science, it's how people look at food, land, families, health, what's right, what's wrong, how we decide that. And from my point of view, it's a great example of how science and science savvy helps us make personal choices and help form public policies, and how science savvy could help us make decisions in the face of uncertainty, perhaps especially in the face of uncertainty. So I'd like to offer 3 themes. One is going to be standards and double standards or dual standards. And you can hear the difference between saying, oh, there are dual standards versus double standards. There's a connotation that goes with double standards that's not a good one. Some people will say we'll have dual standards. Some people will say, well, that's a double standard. Another thing with biotech is to ask what triggers regulations. And that's a big question anytime you have regulations or any ethical or moral structure, whether it's got the backing of the government behind it or not. And finally, as somebody who likes to help people be careful of what metaphors get slung at them and what metaphors they may sling at other people, be careful what you talk about, because we're going to be talking about gene transfer, gene editing, and gene deletion a little bit. But think of the connotations that go with these different things, because what struck me over the last 3 years is how the reaction to CRISPR gene editing has hardly raised any concern at all compared to the issue of gene transfer 30 years ago. And some of you may remember a little book by Mary Shelley, 1818. Frankenstein? Yes. They made it into a great movie. Okay. So, I get to do science, and one of the fun questions to ask people is, what is science? And if you get two scientists locked up in a room, you'll get at least 4 answers. And the next question, though, is how is science different from other ways of knowing? And that's an invitation to think about, wow, what are other ways of knowing? Because as a scientist and an extension person, I get to remind myself, and it's been pretty easy because my dad was a history teacher in high school. There's a lot of different ways that people know stuff. And a lot of different people use a lot of different ways to come to know stuff and come to decide what they're going to do or not do. So the branch of philosophy that asks what is knowable, what is the nature of proof and disproof, is called epistemology. And in the last 15 years or so, when people started using this acronym STEM, S-T-E-M, Science, Technology, Engineering, and Math, I now use this as a little joke. It worked really well because you all died laughing. Here's the irony to me. Math, at least pure math, is over here. Math is deductive logic, and science is empirical. And they're like 179.3 degrees apart. And yet we lump them together as if they're cousins in epistemology, when in fact they're very different. You ever notice how in physics, who wins the Nobel Prizes? The theoretists. And when do the theoreticians win the Nobel Prizes? Right after the empiricists prove that their theory was right. Everybody get a DNA tube on the way in? Yippee-ki-yay. You knew it was going to be a good day. This guy offered me a tube of DNA. First of all, what do you know about the visibility of DNA? Can you see DNA with the naked eye? No. And yet I handed you a tube and it says it's a sample of DNA, and it's from a moderately reputable source. So what is that stuff? Thread? Okay, what else might it be? Yeah. TP? Floss? Remember, I do this with 5th graders, and they're going to be a lot more voluble than you are. Wow, that looks like... Never mind. Okay. So I started handing these tubes out in July of 1995, and I thought people would go, wow, cool. I didn't know you could see DNA. And what people would say is, wow, cool. What is this, really? Like, what do you mean, what is it? It's DNA. It's like, no, it can't be DNA. DNA is too small to be seen. This must just be thread. Out of this pushback comes a whole shtick, a whole paradox. And what is a paradox but a situation where you have two apparently true yet contradictory statements. DNA is too small to be seen. This is DNA and I can see it. How do scientists go about resolving paradoxes? Well, one thing is to start coming up with as many possibilities as you can think of, so you can make this really long list of things that it is, like all the things that you mentioned. I'd also like to suggest that anytime you have an idea, like maybe it's DNA, which is, in the fancy word of seditions, is a hypothesis. And do you remember when you took your course in probability and statistics? If you have a hypothesis, you have to have a... What do you compare your hypothesis to? The null hypothesis. So you in this room, many of you have had a course in probability or statistics. What did Mark Twain say about statistics? What was that?
Figures do not lie, but liars figure.
See, I remember lies, damn lies, and statistics, but maybe it was that one too. So this is a big idea that anytime you have, maybe it's DNA, you should also treat fairly the null hypothesis. Maybe it's not DNA. Maybe it's all these other things. And how do you go about, what do scientists do when they have all these ideas then? They test them. So in your mind's eye, would you please spell the word scientist? And take that second I, and what do you get if you make the second I into an E? Scientist. So if you can remember that, since most of us don't have to do spelling tests anymore or spelling bees, it'll be okay. To me, that's what distinguishes empirical scientists from other scholarly fields. We test our ideas empirically, often through experiments. Now, if I tell you that DNA dissolves in water, how could you go about testing whether this is DNA or whether it might not be DNA, whether it might just be thread? I put some water in there. Like, yeah. But I'd like you to do some Promethean thinking. Who was Prometheus, by the way? He was a Greek god. Wasn't he a mathematician? You're thinking Pythagoras. You from Minnesota? Who here is hoping that the Vikings will go all the way to the Super Bowl and go 0 for 6? Thank you. Okay. Pythagoras was a mathematician, and Prometheus was a Greek god or demigod, depending on which of the many versions you look at. And Prometheus did something you shouldn't do, and he pissed off Zeus. And he got chained to a rock, kind of like our foreign policy for the last 18 years. And the way he pissed off Zeus was he gave great gifts to humanity. He gave fire, and he gave hope, and he gave his namesake. And Prometheus literally means knowledge of forethought. In other words, plan ahead, anticipate. And you guys get that joke. Wow. Tell that to a bunch of fifth graders. Why did he just hit the top of his head? It's called. So if you pour water into your tube of DNA, and if it is DNA, what'll happen? It'll all go away. And then how much more DNA will you have left? And then how many more tests can you run? And how many total tests have you run? So the big deal here is, well, let's, instead of pouring the water into the tube, why don't we take the DNA out of the tube and break it into two, not into two equal parts, but into a itty-bitty part and a big part, because now you're inventing one of the most powerful things ever invented by humans, and that's taking a sample. So you put the big piece back in the tube, you torque that thing back on, you put it in your pants pocket, and you take it home as a souvenir. Unless you came from a great distance, then it's a souvenir. Thank you. And then you get a little drop of wah-wah on the end of your finger, and you rub-a-dub-dub the little piece of DNA. And if it goes away, what have you proven? Nothing. Wow. And you would have any reason not to believe me? I'm here from the government. As the woman noted when I said I'm from Dixon, Illinois, it's like, oh, you're from Ronald Reagan's hometown. Yes, okay. So yeah, you have to believe me. I think if you take the water and it does dissolve, you've proved whatever that white stuff is dissolves in water. What are you going to conclude, though? So let's try logical syllogism. Let's start with the major premise. DNA dissolves in water. That's a true statement because you believe me. Or you can whip out your phone and look it up. Then there's the minor premise. This stuff dissolves in water. You know that's true because you just did it. Therefore, what are you going to conclude? It's DNA. Yeah. So this is what's cool, is that you can have a logical syllogism that has composed of a true major premise and a true minor premise. But for those of us who stayed awake during our logic classes, which does not include me, you realize, well, that's not enough. It also has to be validly constructed. So let's try another logical syllogism. Cotton does not dissolve in water. This stuff does dissolve in water, therefore it is not cotton. Have we proven that this is DNA? So this is the important part. Bob your head up and down and say no, no, no. Okay. Confuses the hell out of the enemy. Okay, so this is news to many people, refreshing to most of us in here. Wow, you can have logical syllogisms that aren't valid, and there's a distinction between proof and disproof. Remember, in geometry class, you do proofs, but in science, we tend to do disproof, and we do not emphasize that enough. We talk about science as, oh, this is the way that we prove things. Actually, we do inductive reasoning, and the mathematicians do deductive reasoning. It's very different. So I can't say to you that I can prove to you that that is DNA. We have certainly disproven that it's cotton and all the other things that are white and fuzzy and that don't dissolve in water. However, if you come out here to the fairgrounds when the fair is going on and you walk down the health food aisle, I'm assuming it's a very short aisle, and you see somebody going like this and offering you a big wad of cotton candy, you have something that is white and fuzzy and dissolves in water. And remember, how many exemptions do you need to disprove something? Just one. So this is DNA, and it comes from a bottle. The bottle cost me $507 for 10 grams. And this comes from salmon. It's highly purified DNA from salmon testes. So the boys gave it up for you. And the other joke is when you just add water, it swims upstream. So this is what I mean by a parable. This is a parable because it's a story that hopefully you can retell. It's a parable based on a paradox. This works for 5th and 6th graders because they've already learned wrongly that DNA is too small to be seen. If I give it to a bunch of 3rd graders, they go, oh, cool. Because they haven't learned yet that DNA is too small to be seen. You got to have an anvil for your hammer to hit on in order to have a paradox. So what are some other ways of knowing that science is competing with? Well, there's reason, and a lot of people think science is reason and reason is science. That's unreasonable. Logic, and there are many flavors of logic: deductive, inductive, strong inference. There's math, intuition. What do you pay when you go to college? Tuition. Do you realize tuition is paying for the learning? Room and board is because you're bored. Instinct, tradition, Tevi, Anybody been to Fiddler on the Roof recently? Authority. What do you call somebody who writes a book? An author. Add ITY, boom. More ways of knowing. Empiricism, which is my favorite. Experiment, which is a specific kind of empiricism. Inference. History, my dad was a history teacher. I think it's a big deal. Literature. Revelation. Prophecy. Mysticism. Mythology. Experience. Superstition. Imagination. Naive theories. I don't know what they're talking about. So now I would like to go on to this question. We started with what is food. Now we're going to go in, or excuse me, what is science? Now we're going, what is food? So everybody remember Wall-E when the little insect crawled in and out of the Twinkie? That was one of the best parts of the movie 12 years ago. Okay, what is food? I'm going to stop here. Please have a conversation with, in teams of 1, 2, or a few. Please come up with 3 foods that come from stuff that's never been alive. Take about 15 seconds. Go. Suggestions. Minerals. I just drove through Iron County last night. It was beautiful. Where do we get iron from? The ground. How does iron get there? It bubbles up from the earth mantle, we think, but actually the banded iron formations that are here in Minnesota and the UP were actually laid down about 2 billion years ago by bacteria. Other ones. Salt? Salt, yep. Except for it's July. It's one of the 3 weeks that's hot. And you go out for a run, and what does your body pump out? Sweat. And then you lick yourself. Oh, boy. And what does it taste like? Yes. So can living things produce salt? Okay. Any other suggestions? This is hard, because a Twinkie is the only one that there is. So this is just a way to remind ourselves that in the fancy words of the biologists, we are obligate chemoheterotrophs. We have to eat chemicals, and we can't fix carbon dioxide. We have to eat things that the plants have already photosynthesized the carbon dioxide and water into hydrocarbon for us. If we were plants, we could just stand outside and we could be autotrophs. But we have to eat other living things. So I put nearly in there because I never know, plus you got to give Twinkies their due. So to keep it simple and not do too many kingdoms, I say the living things are plants, animals, and microbes, and that's what we've been eating all along. And about 10,000 years ago, we being humans, started developing agriculture, and we selected or developed crops, livestock, and cultures. And we select and develop those crops based on traits, and these traits that we're interested in include taste and color and ease of preparing, yield, vigor, nutrition. A trait, as opposed to a trader, a trait to a biologist is a combination of the genes and the environment that the organism is living in. And that's a big deal because if you're a farmer, or a rancher, or somebody that's growing livestock and plants and microbes, that means if you want to change the traits, you can manipulate the genes, or you can manipulate the environment, or you can do both. So if you want to be a plant breeder, you need sources of genetic variation. Where are you going to get those? The whole source of your available genetic variation is called the gene pool. And then you need methods for finding the genes and moving in them and selecting the plants that have the desirable traits, because not all of them are going to have it. And you want to select the ones that have them so they can become the parents for the next generation. Now on the right there is a small picture of the DNA fountain at UW-Madison. So if you come visit me at the Biotech Center there, we can show you that fountain. It's the oldest and largest, most accurate DNA fountain in the world. Trust me. Or don't, and look it up on images.google.com like I did. So genes, can they flow in nature and can they recombine? And a lot of people, this is where things start getting a little squishy and ishy. It's like, because here's this species and here's this species, and you know, they don't mix together. But that's not what we find when we study nature. That genes flow in nature within species, between species. We have lost conjugation. I hate that when I lose conjugation. What will I ever do? Some of the fancy words for the ways that genes flow in nature are transformation, transduction, conjugation, cell fusion, viral infection. Some of you may have noticed that the Google Doodle yesterday was in honor of Har Khurana, who was a professor at UW-Madison from 1960 to 1970. He won the Nobel Prize in 1968 for decoding DNA. He was the second person at UW-Madison to win a Nobel Prize in physiology or medicine. The first person there to do that, I can't bring up his name right now. Anyway, he studied bacterial conjugation. That's why I'm so disappointed that conjugation is hidden. So the big deal is DNA is the carrier of our genes. So that little tube that you have in there has all the genes from salmon. Many copies of it. For those of us in plants, we're very interested in this bacterium from a historical point of view called Agrobacterium tumefaciens. That literally means the field bacterium that triggers tumors. It causes a disease of plants called crown gall, and it is a pioneering tool in biotech in the sense that this bacterium, its strategy is to have a little circle of DNA, and it injects the little circle of DNA into the plant cell, and the plant cell takes up that little circle of DNA, and now you have bacterial DNA incorporated into the plant cell. Ah. Well, if that little circle is kind of like an envelope, what's the cool thing about an envelope? There's at least two things. First of all, you can put something in it. And second of all, you can put the address on it. And where does the envelope go? To where you address it. And what happens? You open it up and the person gets it. So you can put DNA that you want to go into a plant cell into that little circle of DNA from Agrobacterium, and it'll deliver it for you. So, here's Paul Berg from 1972. He won the Nobel Prize along with Cohen and Boyer in 73. These are the three people who were given large credit for the whole idea of recombinant DNA technology. And what this means is, in 1970, if you were a corn breeder, your gene pool was corn and its close relatives. In 1973, at least theoretically, what was your gene pool? Everything. Became a gene ocean. And that's a big deal. Okay? I have colleagues that do work like I do, and many of them will frame this as this is a tool, but it's just a tool like all other tools, and that may be or not. But I still think this is one of the most powerful tools ever invented. However, because it's a very powerful tool, does that mean it's riskier? Well, that's the question. Just because I'm willing to say this is a very powerful tool, more powerful than some other things. but that doesn't necessarily follow, in my analysis at least, that it's riskier. So here's some milestones in the history of biotech and food. 1983, 10 years after the technology is first published about, we get transgenic plants. Transgenic meaning a plant with at least one, maybe 2 or 3 genes from another species. And that could be far, far away. And they used agrobacterium, the crown gall bacterium. And in 1986, there was the first field test of transgenic plants. And the first field test of transgenic plants in North America was in Wisconsin, in Middleton, Wisconsin. And guess what crop it was? A much more important crop than corn. Tobacco. Now, you folks living up here in the central part of Wisconsin don't realize, unless you came from over by Viroqua or down by Verona or over by Edgerton, that tobacco is a very big deal in Wisconsin, still is in some districts. And tobacco is a very interesting crop to grow in the greenhouse. I did my PhD research on tobacco mosaic virus, so I know actually how to grow tobacco and how to rub it real nice and gentle, so it gets a virus and all this stuff. And it was the white rat of plant biology from about 1925 through about 1995, when it was supplanted by Arabidopsis, the little watercress plant. This is another one that's near and dear to Wisconsin. How do you get cheddar cheese? Get yourself a cow. Preferably one that's giving milk. So you get yourself some milk, you add bacteria, you add an enzyme, you add some salt, and because this is Wisconsin, you add orange. Yeah, annatto. So when you go to other foreign countries like Oregon or New York, what's the color of the cheese, the cheddar cheese there? Oh, what is wrong with those people? Well, the enzyme that you add is a thing called rennet, and rennet is a gamush, and the active ingredient in rennet is chymosin, and chymosin is a protein, and this protein is an enzyme, and it cleaves particular parts of the milk so that the milk coagulates. It clots, it becomes like yogurt. And then you rake a wire through it, and it separates into curds and whey, and you let the whey go away, and you compress the curds into cheese, and you let it age, and then you eat it. Where does the chymosin come from? Comes from the calf, the left calf or the right calf. It comes from baby calves. Now, not anymore because of semen selection, but it used to be about half of all baby calves born in Wisconsin were bull calves. And what can you do with a bull calf? Well, you can milk them once. So a lot of times people eat them, turn them into veal calves real quick. So if you kill them before they're 10 days old, you can extract the contents of their stomach, and that's where we get the rennet, and the rennet is the enzyme. Now, because that's an enzyme, it's a protein, that means there's a gene for that. And if you can find the copy for that gene, what can you do? You can give it to a bacterium. And then that bacterium you can grow in a big vat. And it and all its buddies will make chymosin. And now you can have chymosin that is not made by a calf, but by a bacterium. Okay, very controversial? No. Yet at the same time, we had bovine growth hormone. Similar thing. Bovine growth hormone is not a steroidal hormone like testosterone, Bovine growth hormone is a protein hormone, which means there's a gene specifically for it, which means you can find the copy of that gene and give it to bacteria and grow it up and make bovine growth hormone. And then you can inject it into the tail head of a cow and she'll give more milk. And I'll get a job. Because this is very controversial. Even though the technology was indistinguishable, the whole outplay of it is very different. Flavor saver tomato. Give a gene to a tomato to make it slow down the softening process so that you can ship a red ripe tomato. And then transgenic field crops were approved in 1995 and first grown in 1996. And then 4 years later we had transgenic papaya, transgenic potatoes, and golden rice all announced. And of those three, from nearly 20 years ago, only the papaya is still produced in the marketplace. So we've already talked a little bit about how important it is to think about science and the powers and limits of science. And in science, statements about what we know or what we think we know. And then we have words like natural, which is a virtue. That's why it's splattered all over almost every food product that you buy. And of course, nobody puts, hey, it's artificial on their food. So now I'd like you to think about spin on words. I always use BGH, even though I'm in biotech, and most people who are in biotech use another term called BST, which is bovine somatotropin, which is the official fancy-dancy term for it. The reason being, the people who are opposed to BGH like to use the H word, which is bovine growth hormone. And the people that are for it like to say bovine somatotropin because nobody's ever met a somatotropin. Are they the same thing? Yes. In baseball, if you're a batter, is it very important that you can pick up the spin that the pitcher is putting on the pitch? If you're a baseball coach of pitchers, is it very important that you are able to train the pitchers to put different spin on their different pitches? So now I'd like to compare these two very different things, vitamin D and steroidal hormones. How many of you would like to have a steroidal hormone put into the milk that you buy at the store? How many of you think it's a pretty good idea to put the sunshine vitamin, vitamin D, into milk? Okay. Does anybody know what vitamin D is biochemically? I can just hear Oprah now. I'll never drink a cup of milk again. So this is a pretty big deal when I let people know that vitamin D is actually a steroidal protohormone because they're like, whoa, that's bad. Steroids are bad. And this is, again, a classic example of how important spin can be. So spin is really big in what terms we spin and gin and how we use them. There's two bad ones here and one good one, if you're for biotech. Frankenfood is the bad one, Terminator's the bad one, and then golden rice, vitamin A rice. Okay. So what are some methods that humans use to genetically modify foods? For plants, we've got selection and breeding and cloning and grafting and hybridization and mutagenesis. Tissue, whoa, there's just a boatload. How many of these have you heard of? You know, there's got to be a couple of plant breeders in here because it's the central part of Wisconsin. Okay? Now this last one's pretty interesting because now we're into gene editing, and that's the one, like I said, that's in CRISPR gene editing. Okay? Keep in mind, those are things, with the exception of transgenesis, that are largely unregulated. This is the background that transgenesis came into in the 1980s when it was being developed. And to this day, you can do almost all of those, and the FDA and the USDA and the EPA don't care. So now I'd like you to think about a spectrum of values about food. I was a Catholic altar boy, and we have holy, 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 and the Jews have Kiddush, 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 if I said that correctly. And from that, I've like to use this idea, there's a spectrum of values about food: wholesome, holistic, and holy. Wholesome asks, is this food safe and nutritious for me and my family? And a food can be wholesome and yet still be loathsome. So for example, in Wisconsin, we've had the long-running argument since the 1890s about oleo. And loathsome has nothing to do with whether it's wholesome. It's just disgusting. And then there's ethics versus squeamics. I did not invent the word squeamics, but I like it because squeamics gets at the idea of ethics has to do with, wow, character or customs. But squeamics are things that make us feel squishy and unhappy. And that's why I have the escargot there. Because I've been to France several times and I still can't eat a snail. Even with relish or without. And then holy is, of course, halal, kosher, eating brisket on Friday and Lent. Science has no bearing on that. So here's some criticisms of recombinant DNA technology. It's a perversion of nature. It could be poison. It's promiscuous and could spread from field to field and livestock strain to livestock strain. It's all about profit and not about commonwealth. It's about power and who controls what's planted or what's cultivated. And then the one that I add is it's also about the nature of proof and how do you go about establishing that a new technology is as safe as, or even safer than, if required, an older technology. I have to thank Monsanto Corporation. Because of them I have a job. And they're often accused of this word on the left of profiteering. But my dad being a high school history teacher, I also think there's another way to spell it, and that's profiteering. And does anybody know where this is? John Brown, and anybody know where it's hanging? It's a mural in the State House in Topeka, Kansas. This is by John Stuart Curry, who also did several murals, including one just like this, the central figure being a freed black slave that's in our law school building at the Madison campus. Yeah, that's a rifle in one hand and the Bible in the other. So is biotechnology safe? That's not a bad question, but I wouldn't mind asking a tougher question, because is it safe is a yes-no question, and I really can't answer that as a scientist. If I can get you to ask the question, is biotechnology as safe as other methods of genetically modifying organisms? Then we can do a comparison. And humans are built to compare. And it implies that both or any of these technologies has risks that go with them. So remember, these are some of the things that people have been doing all along. Many of these have been done without really extensively looking at how risky they may or may not be. But there's this thing called the test of time. You've heard of the test of time. It must be okay because it's withstood the test of time. And what is the test of time? Nothing. We bury the dead ones and don't know why. Okay? So, which of these are natural and which happen in the absence of the hand of humanity? And does it matter as a point of risk? So, we're going to now look at proof and standards of practice of proof. I heard this from a German professor 25 years ago. He said, remember, there's a difference between the English point of view of proof and the continental point of proof. And by continental, he meant the peninsula known as Europe. In English law, unless something is prohibited, it's permitted. Newsflash, Wisconsin people, we live in a permissive society. Unlike the continental system, unless it's permitted, it's prohibited. So if you've noticed, there's a little tension between Britain and the continent. So the next big question is, should regulations be based on the process that's used or the product? And one of the big questions is, well, where lies the risk? And for 20-some years, and then reaffirmed 2 years ago, the conclusion by the National Academy of Science of the United States is the risk is in the product and not in the process, and therefore we should assess the products and not the risk, assess the products and not the process. Is that how our system is set up? No. And then this leads to the Jesuit question, what should be compulsory, what should be permitted, and what should be prohibited, and on what grounds? Now, what are we going to compare to? Should the safety of genetic engineered crops should they be as safe as all the other methods that we use, or should they be higher? And if higher, how much higher? And now we have this question about, well, what are we going to do about gene editing? Because gene editing doesn't necessarily involve transfer of a gene from one species to another. So you don't get the Mary Shelley Frankenstein vitalistic weird thing going on. Well, we're just going in there and doing a little word processing. So this goes back now 31 years. And then 28 years ago, 29 years ago, the National Academy came out with this report, not only to conclude comparable comparison should be the way to go, they introduced the term genetically modified organism. And since we hear this so much, I would like to point out this is a great example of a flip-flop in how a word is used. That term originally meant what it means. It's a very broad term. It means an organism that is modified genetically. And within a few years of this report coming out, the meaning of it and the use of it had completely flipped. It meant only organisms that were genetically engineered. And how that happened, I don't know. I watched it happen. I was like, why are you using this term this way? And so today when you go buy orange juice, it'll say GMO free. Well, oranges are genetically modified by many, many ways. It just means they're not transgenic. Okay. So crops modified by these new methods should pose no risk different from those modified by classical genetic methods for similar traits. In other words, assess the product, not the process used to get there. And this continues to be something that people are concerned about. And so the National Academy keeps having big meetings and big reports come out, and the latest one came out in May of 2016, and it found the same thing. There's no substantiated evidence of a difference in risk to human health between currently commercialized genetically engineered crops and conventionally bred crops. That's the food safety issue. It also found no cause and effect evidence for environmental problems from the GE crops. So it's coming up on 9:42. I've been talking for close to an hour, and I want to leave time for questions. So here we are. Is it safe versus is it safe enough? If you drive 66 miles an hour when this sign is out there, can you be pulled over? Can you be ticketed? Can you go to a judge and say, this makes no scientific sense, it's barely any more dangerous than going 65? you can try to do that, yes. Okay. So keep in mind the difference between is it safe versus safe enough, and sooner or later somebody has to draw a line somewhere. And science can assess the risk, and we can draw nice little graphs of speed versus danger, but somebody has to draw the threshold. Here and below is acceptable, there and above is unacceptable. So I hope that you will think about these ideas because they're going to bear on how we look at gene editing through CRISPR, how we're going to look at things like, whoa, what do we do when we have livestock that's transgenic? What are we going to do when we have salmon, like last year approved transgenic salmon after 22 years of review? Because this is, you'll notice all the data slides that I presented to you. This is a presentation on epistemology. It's talk about how do we go about making decisions and what kind of ways we formulate standards and comparisons. And so the key thing for me is we've been eating transgenic crops since 1996, and there's no documented illness from it. That's no small thing. Does that mean that it'll always be that way? I don't think so. But does that mean it's about as safe as other methods that we've used? I think you could argue that. And so this helps us, I hope, use it as a model. That's the alarm saying it's 9:45 and stop talking. You need to go to the bathroom? Say that again, please.
You have a lot of stuff to tell us there, but not much time. That's what they're.
Oh, no. This is all the really good stuff. Okay? The really good stuff is about to happen because I'd like to open this up for questions so that you can make comments or questions or give your point of view. If you have historical
Anybody over here?
stuff to
You have a question?
comment on, that'd be great.
Questions?
If you've worked in the potato industry in the Central Sands, you might have some things to say.
I have a question about, I went to UW-Madison, science background, and I'm having a problem with like organized religion and its influence on science, especially like a lot of politicians are getting in there and imposing their belief system. And I think, for myself, I see a lot of folks are kind of holding us back like an anchor, where a lot of scientific breakthroughs that could help, like my mother had Alzheimer's, and now I'm very concerned about research on that, so I've had to deal with that before. And when people come in and say, no, I've decided because of my belief system that you can't do any more research on that, it really bothers me a lot. And I wonder how you feel, I guess you have a religious background as well, how you feel about the role of religion as far as scientific breakthroughs. Thanks for coming.
Well, I hope the questions get harder than that easy one. Yes, so faith versus empiricism, the freedom to practice versus exercise. Remember what does the Constitution say? The word is exercise, not practice. It's very interesting in the United States, the role of religion goes back to the Test Act in England in the 1680s. It's right there at the penultimate paragraph of our Constitution that there shall be no religious test whatsoever for any officer of the United States or the several states. We've got it in the Bill of Rights. It's very controversial. We also have this little thing called academic freedom. And you've got a copy of the plaque right here on your wall about 40 feet from where 40 yards from where we're standing about the continual and fearless sifting and winnowing. On the one hand, we have to be able to respect what people's religious beliefs are, and yet on the other hand, to the extent that they would stymie free inquiry is something that academics have to take very seriously. We're coming up on the 20th anniversary of the announcement of human embryonic stem cells by Jamie Thompson at UW-Madison. I think that's one of the classic examples of the role of religion, especially the abortion issue and the federal funding. That issue was primarily focused, if you remember, in August of 2001, because nothing was really happening in the summer of 2001, except if you look back at the papers that day, every day there was an article on what is George Bush going to do about federal funding for human embryonic stem cells. And then this little thing happened on September 9th. No, 9/11, September 11th. Okay. And people have no real good memory anymore that the dominant national issue in the summer of 2001 was this interplay between religion and federal funding of research. So there's this issue of elected officials feel like they should be able to control where the federal funding goes. That's one thing. They also feel like they should be able to regulate whether that's funded by state funds or not. And they can, and they do. And there are states where research on human embryonic stem cells, for example, is illegal, regardless how it is funded. So this becomes a conundrum because researchers have feet. And as Ronald Reagan pointed out, you can always vote with your feet. And one way that you can depopulate a state university of really excellent people is to impede their research in ways that other states are not impeding their research. So those are some of the costs and things to consider when people want to make laws and regulations on funding and performance of research.
I wonder if you could address the Roundup Ready corn and the lack of monarch butterflies.
Could I address the issue of Roundup Ready corn and the lack of monarch butterflies? This is a very big deal. Monarch butterfly became the, I think it's fair to say, the poster child of the anti-biotech movement about 15 years ago. BT corn produces a protein that kills caterpillars, including monarch caterpillars. And if it's in the pollen, then the pollen can shed all over and it can fall on the milkweed in the rows, not the rows, the fence rows. And if the caterpillar is there at that time when the pollen is falling, then the caterpillars could die. And that's the scenario that people rolled out. It's a very convincing scenario. And then when people took the 2 or 3 years to do the research that was triggered by a 2 or 3 paragraph scenario, it didn't pan out quite so well as to how much BT is in the pollen, how much pollen is actually falling there, when the caterpillars are there. But it's a really good example of how people could reasonably say, oh, here's a scenario, and then it takes years to actually go about testing it. And if that had been a documented case, I wouldn't have been able to show you what the National Academy said just last year, that there is no documented case of harm from the environment.
Is there any food that you do not buy for your family, assuming you like the product?
For some reason, the word broccoli comes to mind. I'm not a child of the Depression, but I'm a child of the children of the Depression, and I am not a good spokesperson for the current foodie movement. And I'll just be really upfront with you. My dad did all the grocery shopping. It was all about getting the most food for the least money. It drives my wife nuts. It's not the only thing about me that drives her nuts. So no, I don't worry about that. Do I buy organic? No. Does my wife? Yes. I will buy organic stuff when it's the cheapest or the best value, and sometimes it is. But I'm not a great spokesperson for a very food conscious point of view of buying stuff. And of course, like I don't even buy brand names. I wouldn't even bought a Twinkie. It would have been one of the Swiss Miss versions. So living in Madison is very interesting because Madison is a big foodie place, and you have a lot of scientists, a lot of very well-trained scientists, and it's interesting having conversations with people who are highly trained, and yet they also have very profound concerns about food, and they choose to say, well, I'm going to only buy stuff at the farmer's market, and I know my grower, or I know my farmer, or however you want to look at it. And I'm fine with that. That's everybody's choice, and I get to go to Woodman's and buy what I want to buy.
Could you explain exactly what golden rice is?
Yes, it is rice that is gold. So rice doesn't have, as I understand it, did not have a vitamin A pathway to get it into the seed, at least. And it was, if memory serves, 5 different genes that Ingo Petrikis and his group in Switzerland had to get into the rice in order for the rice to start making vitamin A, or at least the beta carotene. And so that was quite a feat. It'd be quite a feat today. And 20 years ago, in 97, 98, 99, it was a decade of work. And the reason for vitamin A rice was to address the issue, wow, we have all these crops that are for agronomic traits, for farmer traits, resist insects, make it easier to spray herbicides on crops. Why don't we have something that has direct benefit for the eater of the food? And so by saying, all right, we're going to address this big issue of vitamin A deficiency, which kills millions of people a year around the world and blinds millions of others. And there are other solutions to vitamin A deficiency besides genetic engineering. But if you're a genetic engineer, then and you're going to work on plants, you might as well take a challenging prog, and this is what Ingo Petrikis' approach was. So that's what vitamin A rice is about, golden rice, and it's called golden rice because if you have enough vitamin A, beta carotene being expressed in it, then it'll turn yellow, faintly or more pronounced. And then the big question is, how does it taste? How are people going to respond to it? How does it cook up? When it cooks, is it still active? So the irony is, there are also golden corn projects where you're trying to get higher concentrations of vitamin A expressed in good old corn. And because those projects are using traditional methods, they aren't nearly as controversial as Ingo Petrikis' golden rice. Of course, they don't get all the positive buzz either. Ingo was the first person that I read back in 2001 where he very forcefully stood up and said, we tried to address all the ethical issues that people placed before us, and I believe we have. and yet they still will boycott it. And I believe the people who are boycotting it are not boycotting it anymore with any ethical basis, but because it meets their political and fundraising needs, and therefore the moral high ground is no longer theirs. You may have read last week in the New York Times a similar article from the president of Purdue University, who used to be the governor of Indiana. And he made that argument. If he had been a scholar, I think he would have cited a guy like Ingo Petrikis, but he somehow overlooked that the same argument had been made in 2001.
I have a concern with some of the genetics that's been done, or the modifications that have been done by Monsanto and some other companies, where they will modify a product like the corn seed that can only be grown with a certain way, and it's leaving out the small farmer, especially in Central American countries, where they can't afford the whatever Roundup Ready corn or whatever it might be. And I question, where does ethics come into that type of situation?
So on the question of who the intellectual property of the seed and how do small farmholders, small farmers in poorer countries benefit from this? The Ingo Petrikis case is an interesting one because one of the things Ingo had to do is get permission from about 30 different entities, including Monsanto, so that he could get licenses to use without having to pay royalties, all the genetic tools it needed and had patents associated with them, so they could get those 5 genes all lined up and get them to work in rice. The other alternative is to get the people that have the intellectual property, the patents, on specific, either the genes themselves or on the processes that allow you to engineer the plants, to provide similar licenses so that people working in third world countries can do the research and deliver the products and the farmers can use them. Most of the issues that I've heard, and you can correct me if you would like, with Monsanto and seed saving have been with soybeans. Seed saving isn't a big deal with corn because what are most lines of corn that we grow in the United States? They're hybrids, and so you don't save seed anymore, and haven't since the 30s when hybrids came in. Soybeans, on the other hand, are inbred, so you can buy Roundup Ready variety of soybeans, plant it, harvest it in the fall, and send 10% of your seed to the elevator and save 10% of it and plant it next year. And will it be Roundup Ready? Yes, it will be. And will you be in compliance with the license that you signed off on? No, you won't. And so you can end up like Percy Schmeissler in Saskatoon, who was a Canadian farmer who saved seed of canola that was Roundup Ready. And so that's probably the most famous case that I know of Monsanto enforcing its patent position on, in this case, the Roundup Ready gene in a crop, Canadian canola. Still controversial to this day, but the United States Supreme Court voted 9-0 to uphold those kinds of patents. And the U.S. Supreme Court doesn't vote 9-0 very often. And so the law, at least in the United States, is pretty clear. The fact that something is legal does not necessarily make it ethical, and you could ask Dred Scott about that. So I think the onus is on the companies when they have intellectual property to say, why would you impede other people from using this in places where you can't make money on this? And that was the argument that Ingo Petrikis was making 25 years ago, and successfully did so that the rice could come out 20 years ago. Although it's still not commercially available that I know of. Does that respond to your question?
We want to thank Tom for coming today. He did such a great job. It was very interesting. Thank you so much. Thank you.